So the very first step of gene expression, in order to use gene's instructions and then make a protein, is transcription. Transcription has two differences in eukaryotes versus in prokaryotes, and we're going to see how it's different in a second. So transcription in eukaryotes, we have the coding strand, and then we have the non-coding strand. Why coding strand and non-coding strand? So the DNA has two strands. Coding strand is the actual instructions that DNA wants to pass on. Non-coding strand is also called the template strand. It's also called the antisense strand, and it's also called the minus strand. So why does it have so many different names? The coding strand is the code that you want. Non-coding strand acts as a template. Why? Because if you notice, the non-coding strand, when it base pairs to the mRNA is going to look exactly the same as the coding strand. So that's why non-coding strand is also called template strand. And these are just two other names that I'm not going to explain that you can call the non-coding strand. So if you think about it, if the non-coding strand is called template strand, that means the coding strand can also be called non-template strand as well, opposites. So you have DNA is split apart by topoisomerase and helicase. Topoisomerase unwinds the DNA. Helicase separates the DNA. This is covered in the past video about DNA replication. So once helicase opens it up, now RNA polymerase can come and build the mRNA. So RNA polymerase makes mRNA. How does the RNA polymerase know what part of the DNA to attach to start making the mRNA? It attaches to a section called the promoter. So promoter is DNA, and that's the section that the RNA polymerase is going to attach onto so that it knows where to go to start building the mRNA. Is there only one promoter in the DNA? No, there's going to be many, many promoters in the DNA, and there's going to be a promoter right before each gene so that the RNA polymerase knows this is where I'm going to start building the mRNA. So next word there is number 5, upstream and downstream. Right? So upstream just means closer to the 5' side. Downstream just means closer to the 3' side. So if you notice, RNA polymerase is actually going to follow the non-coding strand from 3' to 5'. When it follows from 3' to 5', it can build the mRNA from 5' to 3'. The RNA polymerase can only add new bases over at the 3' side. So it makes sense that it builds 5' to 3', but it's going to read these instructions from 3' to 5'. And even though it's reading it backwards here, it'll actually be in the correct direction because you want the mRNA to look the same as the coding strand. The last thing we're going to talk about for transcription is enhancer sequence. So enhancer sequence is just part of the DNA that's going to cause the DNA to bend. That way you can have more transcription of a certain part of the gene. So here the RNA polymerase is attaching here, and we're going to have transcription this way. Enhancer sequence is going to allow the DNA to bend, and so that the RNA polymerase will transcribe more of a certain part of the DNA, and we'll have more of that mRNA. Now, as I said before, this is transcription eukaryotes. In prokaryotes, example bacteria, there's no nucleus, right? But also on top of that, behind a promoter area, there's actually more than one gene. So in the last picture we just talked about, before each gene, right, there's a promoter so that the RNA polymerase knows where to start. For prokaryotes, right, there might be more than one gene that's right after the promoter. And so when RNA polymerase attaches onto the promoter, it will get the instructions of more than one gene into that mRNA. Now, there's a new word here, operator. So in eukaryotes, there's more than one way to stop transcription, right? So if the helicase doesn't work or the topoisomerase doesn't work, then we can't even open up the DNA, right? There's a lot of other transcription factors that can start and stop transcription that we're not going to talk about in this video. In prokaryotes specifically, part of the DNA is called an operator. So there's the promoter and then there's the operator. The operator can stop the RNA polymerase from continuing to do transcription if a repressor molecule attaches on. So you can imagine it's like a blockade, right? So it's like a barrier. The RNA polymerase cannot continue reading the instructions and building the mRNA if there is a repressor molecule attached onto the operator. An inducer molecule is another type of molecule that could remove the repressor molecules. That way, the RNA polymerase can then continue reading the instructions and do transcription like normal. Finally, a transcriptional activator molecule would then be able to cause more transcription because it helps the RNA polymerase attach to the promoter better. This entire section is called an operon, the entire section being the promoter, the operator, and all the genes after that. That would be considered an operon. So operon, once again, is just describing the promoter, operator, and the many different genes that are after that promoter. That entire section would be called an operon. So would bacteria have more than one operon? Yes. But remember, each operon can contain more than one gene inside of it. While in the eukaryote, it's usually one gene right after the promoter.